Welcome, ladies and gentlemen, to the Agger podcast, the first podcast that we're hosting live. And today we are with a very special guest, and that is Matt from Shooting the Soil. Hi, Matt. How are you?
Hi, I'm great, guys.
That's awesome. And then, of course, we have Royce here. And today we're just going to be talking about microbes, microscope analysis, and just general philosophy about farming, gardening, and all things good like that. So I think we want to start this episode off first with a little bit of an introduction to Matt and how he even got into this. So Matt, do you mind telling us what kicked you off on this microscope experience?
All right, so I've always been a home gardener. Growing up, my parents had a garden and everything like that. And then I got big into the organic gardening and, was on the whole bottle kick and everything and, having some pretty good results. But then this was like 3 years ago, a buddy came over and was like, hey, you should come over to my cousin's place. He's doing this whole living soil thing. And at the time, being so into the bottle kick, I just heard, oh yeah, not using pesticides, not using chemicals, using organic fertilizers, same thing. And then fast forward to actually just the beginning of this year, I stumbled upon an Elaine Ingram video, and I just was blown away. I had no idea. It's like, how did I miss this? It was such a humbling moment, as far as just getting a bigger understanding of things. And I actually called up my buddy. He got me on the phone with his cousin. We talked for like 3 hours one night just about living soil. And ever since then, just been hooked. And I just started a class with Matt Powers to be certified in regenerative soil. And 7 weeks into that 20-week course.
Very interesting kickoff there. And I have just a real quick follow-up. What, in your definition, if you could make your own, what do you consider to be living soil?
Just soil with biology, not being dirt. And, it can start off small, but you'll notice the difference just in your soil texture. As that biology starts to grow, they're the drivers. They're they drive the car as far as the soil. The plants help out, but the biology is what really creates the soil. So if you have low biology, just have a couple of microbes, just mostly coccyx, very few shapes and diversity. And then as your soil advances, you get more and more and more until you become almost fungal dominant, and then have just as much fungi as bacteria. And they all play their part in the soil food web.
So the main kind of groups of microbes that you're looking at are fungi and bacteria, and you're saying there should be like an optimal balance of those.
Yeah, pretty much. Like as the soil advances, you'll see more fungi. For instance, fungi kind of like a lower pH, and the plants will drive the pH lower. They like, they'll drive it down to like a 6.5, 6, even lower than that, depending on the plant. And basically actinobacteria will compete with fungi for pH. So that's a big tip when you're looking at your, if you see a lot of those weird, funny looking strands, they're most likely actinobacteria strands and not fungi strands, because you don't see those nice, septas, the nice separations in the hyphae. They don't have a nice color. They're usually clear. And what they'll try to do is push the pH higher and try to outcompete your fungi. So that's just one step in the whole advancement, and just one thing that you can see where two species will compete with each other. So
what would you consider like the ideal balance of fungal to bacterial, the fungal to bacterial ratio?
It's pretty much 50-50 that's the goal, but I don't focus too much on that, because everything that we're learning, My dog's over here playing with a squeaky toy or a squishy toy. You're crunching in the background. Go get it. But alrighty. Anyway, fungal-to-bacteria ratio. So I don't focus on it too much. Could you play with a different toy? All right, whatever.
We don't
I
really
hope
hear it that much.
That's not too loud. It's
It's
her
not.
crunching in the background, is it? All right. But anyway, so with the whole endophyte system, how the plants can suck in bacteria, and it's a huge resource for them, and they can actually be eaten inside of the plant and in the roots, spit back out, and then regain nutrients, go back in and repeat the whole cycle, and actually be nitrogen fixers. We can get deeper into that if you want, maybe later on, but just that whole thing like points a lot to, hey, the bacteria is really, really important, maybe just as important as the fungi. So yeah, you want that 50-50 ratio, and that's your goal to advance your soil. But at the same time, like, you want to focus on the bacteria too. That's my one point and one thing that I'm really learning from Matt. And this whole discovery of this, rhizophagy, endophytic, relationship with plants.
I definitely
So-
agree with that.
Do you think I could just pause right here and ask, these plants are eating microbes? Does that mean plants are not vegetarians?
They're omnivores, all plants. All plants are omnivores.
That's really interesting.
Yep.
I'm sorry, Rob, to cut you off there.
No, that's a great little. I've never thought of it like that, to be honest.
That kind of blows my mind.
So hey, if you want to eat plants to be vegetarian, maybe you're not so vegetarian yourself, because the plants themselves aren't fully vegetarian either.
Yeah. Well, if they're grown in dirt, you don't have to worry about it. If they're grown with pesticides and whatever, then there's no microbes in the soil. Then yeah, all right. Yeah, they might not be omnivores in that sense. You're safe there. But
Interesting.
really, your gut needs that bacteria and all that thing for you to be healthy. Just for the plant to be healthy.
Right. And
They
actually,
work
I
very
find a
similar.
lot of similarities between like your health and like the way you balance like your own nutrient values with like how to balance a plant's nutrient values. I know there's a great
Yeah.
little app called My Fitness Pal that I use to count my calories, and I feel like plants could definitely use something like that. Got to count that bacteria and nutrient uptake.
Yeah, exactly. Yeah. And they help break things down. We get more energy for our buck as far as the food we eat, if we have the right microbiology in our gut to break things down. And it's the same thing. You get more bang for your fertilizer, or whatever's in your soil, if you have that biology to
So what are the
properly
most
unlock.
like common and what are the most common bacteria that you find?
So really you can only see the shapes using a microscope. You can get a little bit deeper, but not much. Like, can you see a little tail? But mainly the three is coccyx, basilisk, and spirilla. So spirilla, little wavy guys. Basilisk are pill-shaped, and the coccyx are round and football shaped.
I feel like when I look at the microscope, I see a lot of coccyx more than anything else.
Yeah, that's the most common type that you're gonna see. Yeah.
So do you want to talk a little bit about your microscope setup?
Sure. So the difficult part about my setup is I tried to put a full frame camera onto a inexpensive microscope, or I don't think it matters what microscope you put it on. But if somebody just wanted to get, a regular setup like this, I would highly recommend either getting a crop sensor DSLR. You're going to have a lot easier time getting it looking right, or just one of the microscope 4K cameras that go right on top. But since I already had this thing, I want big, I want that full frame resolution. And this microscope does pretty good delivering to the full frame, but I really need to step it up to take full advantage of this camera. And the next level, like I was talking about before with you guys, is it's like $4,000 range, to get into the next level. But with a $300 microscope, you can see pretty much what you want to see for being a home gardener, unless you want to do art and really dive into it and make a YouTube channel out of it. You don't really need the $4,000 camera. You'd be just fine with, any of these like crop sensor DSLRs, or just buying the 4K microscope camera and throwing it on a, $300 to $600 microscope, and you'd probably be more than happy. But yeah, to really get the high definition and, be able to like zoom in for days, since you have that 4K resolution, that what's I want to do, you need the better, focusing and depth of field capabilities that you don't really get in $1,000 or a $2,000 camera. You really need to step it up,
Right.
to the to the big boy, from what I've heard. And to get the the epifluorescence and the backlighting and everything like that. So
Yeah, I can't really talk much. I have like a little $50 camera here. Doesn't do too much, but it gets the job done to just get
those that just makes it simple, and it works, and it's built for it, so it fits the kit. Like when I hooked this thing up to this microscope out of this box, I had a little circle in the middle of my big sensor. It was like all black, and I had a little circle. So I had to buy almost I think it was like just under $700 to get this other lens to then make the the light and that little circle fill the frame. That was the hard part about it. Is this little adapter thing, which I then had to jerry rig. This is a 3D printed part to actually get the focus and everything matched up correctly, because I didn't quite have enough height, and I was getting a lens flare. So I had my friend print me a little 3D printed mount so it could be up a little bit higher. And now it's working pretty good. You can see in a couple of my earlier videos, if you go onto my channel, there's a bright spot in like all of my videos, and in like the last 3 or 4, there's no more bright spot in the middle. So I figured that out.
It's awesome.
I have to say, from like a non-microscope owner's perspective, whenever I give my samples to Rob and Rob looks at them under the microscope and sends me pictures, I am just blown away by just the fact that there's all this stuff going on in the soil that you can't see just by looking at the garden. Yeah. And I would definitely encourage people to check out your channel to really see, the benefits of looking through a microscope and seeing what's going on in their garden soil, because it's a whole different world, and it's really fun just to see it happen.
Yeah. And I know you do a lot of live streams too. So the audience can always go and ask live questions. I mean, hopefully we get some live questions here, but if not, go check out Shooting the Soil, because I know you're live pretty often. So
yeah, I'm starting to get a little bit bigger. Hoping to get, hoping for it to catch on a little bit. But I've got a couple live, not really any too much interaction live. Hoping for some more, and the coming live streams. I'm up to like 425 subscribers at the moment, so
yeah, congrats.
Slowly growing, but
bigger than us. So all right, so like if you were to like tell someone who wanted to buy a microscope, would you want them to go your route? Like what would you tell them as a novice guy who, say Royce wants a microscope? What would you tell Royce?
Yeah, so I would say definitely, this is a Swift 380T. I think it costs like $275 on Amazon, and it works great. But I wouldn't bother with looking through the eyepieces. Like, personally, it just wasn't for me. And once you have it on the screen, there's just so many more aspects. So I would buy, Swift actually sells a camera, like a 4K camera. You just put it right on the top, plug it into your computer, and you're good to go. Like, that simple. And it's gonna look good, and you're gonna be able to share samples with people. People are gonna be able to look at it and give you advice, or you're gonna be able to say, hey, what's this? And somebody else might know what it is. And, that's the best way to just gain experience, because if you're sharing eyepiece with somebody, it's like, oh, what do you mean, that thing top right or top left? When it's on your screen, you could just be like, no, that thing right there. That's the biggest thing. So I would definitely say camera, hook it up to a laptop or a computer is the way to do microscopy.
And this way too, you can, if you see something so interesting, you just be like, quick capture,
Yeah, just take a picture.
Because that's the problem I have. I'm always in the eyepiece.
Share it
Because again, my
online.
camera's not the best, but they're actually, I mean, I kind of enjoy looking through it with the little binoculars and feeling like I'm like just like staring at these microbes and the bacteria and all the organic matter, and like feeling like I'm actually there. It's a little bit immersive for me, whereas like on a screen, I feel like, oh, it's just like a video, but
Yeah.
each their own. But I definitely agree though. If like
Get
you
a
were
bigger screen so you're fully immersed in the screen.
imagine it's going to be VR next. We're all going to put VR goggles on and be going through the rhizosphere within VR. Oh man.
I've already been thinking about how I could get like a mechanical stage and control the focus and the slide with like a video controller.
Oh
That sounds cool.
So just sit back here and just look around.
That's great.
Yeah, someday I'll figure that out.
Matt, have you ever seen a microbe eat another microbe live, like on the slide?
I have.
Is
Actually,
it like, does it like tear it apart, or is it just like swallow it?
Just swallows it, and you can kind of see it inside its body. You see, it's like, oh wait, was that just there? And it happened so quick. You kind of have to go back, and it was like, wait, was that little round thing in its body like in the frame before it just went and ate it? And you're like, no, it's not. It's like, oh, it did eat it. So
Weird.
but it was an arthropod eating a little cilia. It was actually I forget one of my videos. It's just one of my 4K dark field videos in my microorganisms playlist. It's just like a bunch of microorganisms. It was actually in pond water that I was looking at, just doing a cool video just to show a bunch of cool stuff and put my friend's music to it. And yeah, it just snags it up.
Yeah, I know. I see lots of so I actually haven't seen an arthropod yet, which is kind of upsetting. I want to see one of those in person, but I've seen lots of nematodes, and I see them all like, I mean, we're going to look at soil aggregates coming later, so stay tuned. But when I'm looking at like the soil organic matter, and there's like aggregates clumped up, and the nematodes are like kind of going all around that. Now they're eating that, right? They're eating like that bacteria and stuff around the aggregates, correct?
Yep, because there's bacteria all over those aggregates. They're that nice golden color that's releasing those, humic acids, amino acids, and stuff like that. Turns it that color, and yeah, they're covered it. They're covering it, and the nematodes seem to know that.
Yeah. So I guess I won't ask that yet. We'll still wait, but I kind of want to stick with the microscope a little bit more and talk about just the different techniques that you can use in microscopy. So I know the most common and the most simple, I guess, is bright field microscopy. And that's just simply big old bright light shining up, and you get your slide, and that light's shining through the slide into the eyepiece, and you kind of see the what would the word be? I guess the shadow. And I actually have an example that I want to show of like IRL per se, bright field microscopy. And I'm really curious about if you guys agree with this. So I'm going to share my screen real quick, and people will be able to see it on the, I don't know which screen I'm sharing. What can you guys see right now?
I think desktop.
Okay. So let me pull this up. All right. So I was just driving to work one day, and it was really sunny out, and I saw a big old sun truck in front of me, but you can't see the details of this truck. So you see that, yeah, it's a truck. It's got a big old bed, probably some like dumpster or something like that. but you can't really see like the details. Like, can you tell me what that license plate is? No. And I felt like that was probably a good example of like in real life bright field. Am I wrong here? I don't know, maybe.
No, that's pretty good. You know, that's a good example of you know, trying to look at a root, something that you know, isn't transparent in the soil. You can't really, all you see is, yeah, no detail, just the edges.
Right. So there's then there's other ways of looking at a of to kind of get maybe more detail and more just image and do you want to explain other techniques that you know about, Matt?
So the only other technique I have is dark field, which I can show you actually it's right here. So all you need is this little piece of plastic here, and it just blocks out the light from the middle. And so what you're looking at the slide is only lit from the side. And then you basically have a black background and everything is illuminated from the side. So you see it's like a whole different way of seeing. It's like a lot more texture, a lot more colors. Fungal hyphae tend to like illuminate in like rainbow neon shades. It's pretty neat.
Yeah, I actually use a little like trick. It's not the most like I guess useful or like the most correct dark field, but my microscope, let's see if I can bring this up here. So you see there's this this slide right
Yeah,
here.
that's where that filter goes in mine that I was just showing you. It just drops in there and that's it.
So what I'll do is I'll like because I haven't had a good filter yet, I'll just like block the light with the slide itself, and it actually works really well.
that can work too.
So I guess the whole point is to like you're kind of skewing the light so it bends in many different ways, because at the end of the day, microscope analysis is really just optical analysis and how you can shape light to come into your observations.
Exactly. When you know, seeing light from right behind, you don't get much depth of field, but then when you come out from the sides, you see all those edges and it makes things look more 3D.
Yeah. And then I know you spoke briefly about epifluorescence. Do you know a little bit more about that?
Yeah, basically that's just having a light, you know, up here on the top of the camera that shines down, and the epifluorescence is just all different wavelengths. And supposedly, once we get everything dialed in, and you know, this is almost based on you know, like spectrometers that use light to tell what minerals are. Essentially, I think we're going to be able to get to the point where we can actually use different light shining down onto a soil sample to see mineral types, and you know, see nitrogen and see phosphorus or copper, or just by the way that they react to the light, just like, you know, NASA detects you know, atmospheres of distant planets.
Now, would you like compare microscopy to I don't know what the correct word is, but I guess astronomy. So like when you're pointing a telescope up in space, I kind of see microscopy as the same thing, because you're only getting such a small glimpse of what actually is the broader view of the world.
Yep, definitely. It's like going, it's just going the opposite direction. It's funny how, especially in the dark field, how it can look like a, you know, you could probably set up just the right image to look like just like something that came from the James Webb, and not actually from space, but it was from the soil.
And hey,
Because
you get to see little
especially,
aliens.
yeah, the bacteria twinkle around like stars. It's really cool.
Can I ask like an off the wall question, Rob? Do you think it's advisable?
Bro, go for it.
Okay, here we go. So Matt, I do a lot of thinking about stuff like this. Do you think it's possible that like we're living in like a tiny world, and there's some like very gigantic, like universal sized thing that could watch us with their microscope? How possible do you think that is?
It could be. I mean, like the universe, it could just keep on going and going and going infinitely. Like, who knows? Like, I don't know. It kind of just like takes my brain to that level where I'm like, do I really want to go this far?
I would say, Royce, if I had to put my 2 cents in, that the people looking with the microscope are probably just in a different dimensional space. So like we're in, I guess, is it 3D or 4D? I don't know if you can count time, but whatever the next dimension up is, which I think is 4D or maybe 5D, they would probably be looking at us through that microscope. So-
That freaks me out even more, Rob, that you added that other dimension to it.
and
Now
you'll
I'm
never
like,
see them.
I'm like positive that someone's looking right now. So thanks for that.
Someone said like the Truman Show. Yeah. We also
So-
have a question here. Why is it called dark field?
Just because you're blocking the light. So it's like the background's all black and you're only lighting from the side. So like, bright field, the shining light through the subject, and dark field is you're blocking the light from going through and only getting it from the sides. So that's the best way that I can, and it's the opposite of light. So they just called it dark field.
Right. And someone else answered the dark field because it's black. Yep.
Yeah.
Very good. Thank you for commenting, ladies and gentlemen. It's awesome. All right. Well, I think it's a very good discussion about microscopes, and we got a little theoretical and maybe a little wooey, but so actually one more thing. What is your preferred optical zoom, Matt? So are you a 4x guy? Are you a 40x guy? Where are you at?
Usually start at the 10x and then go to the 40. So 100, 400. And that's where I spend most of my time. So 400 to see the 100 to scan around for, nematodes. And then I go to 400x to, just check out the bacteria. And then I ditched my 100x, the 1000x aperture, because I just don't like messing around with the oil. So I actually replaced it with a 60x, which gives me, 600x. And that one's fun to use every once in a while, but it's really fine tuning it. The focus is like super fine, and I'm like, the objectives is like, I'm going in between 1.5 millimeters from the slide. It's pretty small working space with the 600x.
Sorry, sorry, I didn't mean to cut you off.
That was done.
It looks like people want to see microscope now. So do you want to pull up your screen, Matt?
Yeah. Let's see. I just got to go over here to share screen. And yeah, that's the screen I want to share. And there we go.
Awesome, I see that.
You see it? All right. So yeah, I just started on this little. So this is the color, fungal hyphae that you want to see.
And
So
right
it's
now
up
we're looking
there.
at it in bright view, right?
Yeah, this is bright field, and this is 400x. So if we go back to, there's the, as far as what the bacteria we see there, and a bunch of little different things wiggling around. Can you guys see my mouse, or does the mouse not show up?
We see it.
You see it?
Yes, sir.
All right. So most of these look like coccyx, but right there's kind of a longer shape one.
And that would maybe be a spirilla.
Maybe be a spirilla. Yeah, it's kind of S-shaped. And trying to see if we see we should see some bacillus in here, because I've been adding a lot of different treatments with bacillus in it. Now, bacillus is commonly found in kombucha, or actually brujun, which is the same thing but just honey. Let's see what else, but a lot of nice looking aggregates in this soil.
I wanted to ask you, Matt, about soil aggregates. And if they're good, they're bad. Like, what are we looking for? And are they good for the garden?
Yeah, more aggregates, the better, and the bigger too. And like over here, you want to see little strands in between, connecting them and gluing them all together. Like, oh, yep, there's a little
Yeah, it's like the big cluster
fungal
of like
hyphae
little
right
grains.
there. Yeah, because that's gonna, it's not only gonna create that texture that you want to let a little bit of air in, so you don't have to, do a massive tilling. But it's also breaking down all those nutrients, making them more plant available. And it's also it's like a glue that's gonna not only hold water in, but it's also gonna hold any other nutrients that you might add to your soil in. So they don't just run off into our, streams and water systems.
So the actual color of these soil aggregates that we're looking at, does a lot of that amber color come from the soil itself, or is that like exuded by the microbes in some way?
Yeah, that's actually the microbes, releasing their exudates onto the minerals to break them down in the form of, humic or amino acids. So that's why, it's funny, bacteria are kind of chemists. They go around and release just the right thing. So it squirts out all these different, different forms of nutrients that they want out of, rock, sand, and silt.
Wow.
So like the clear things are more, what you would consider a piece of sand or a piece of, or, salt, just not something that's not being broken down. And it's funny when you put a lot of the, and this soil before I adapted, why you see a lot of this clear stuff all mixed in with the nice aggregates that are now starting to form, was probably because I was using salt-based organic fertilizers for the 2 years prior on this soil before I switched to living soil at the beginning of the year. Yeah, and since then, like the brown aggregates have tripled, if not more. Oh, look, we got an amoeba. This is a testate amoeba, I believe.
How can you tell that?
Just because he has this kind of foot over here and he'll like grow and shape and move around. And I'm pretty sure I've seen them called testate amoebas a couple of times. So I think he's just holding on to a little piece over here, but they'll usually hold on to something and then go out and try to wrap themselves around things and pull them into themselves. They might change shape a little bit here if we look at them for long enough.
I was going to mention before that I heard that in like the soil food web, the most efficient eaters are the bacteria. And so they're going to live and die and all the nutrients kind of stay in that cycle. But the larger ones, like maybe the amoeba, are kind of inefficient eaters where a lot of the things that they're eating, a lot of it's just kind of going to get pooped out and it's going to be available for plants then.
Yeah, because they're pretty much going to eat the bacteria and the other things, and bring them into them. But, so they're kind of a predator. But yeah, when they, just like when they poop out, it's fertilizer essentially.
And that's like a slow release fertilizer too. Nature's best slow release.
I'm pretty sure it's, it's slower than a lot of the others. I think, like a nematode is a much faster predator. You just see them going around, tearing through bacteria, the bacteria feeders, fast movers, eat a lot more and, poop out a lot more.
So I know nematodes are probably the most popular or most popularly known microorganism. What would you say are your favorite least popular microorganisms, or just favorite microorganism in general?
The tardigrades, pretty cool. The water bears.
Yeah.
I found one of them on the Salt River Marsh by my house, but I haven't found one in the soil yet.
Yeah, I don't think I've ever. I think they're pretty rare to find in the soil. No.
Yeah, but they could be in both. They can live in, they're what is it? What's the term? Live in oxygen and lack of oxygen environments. They are I'm drawing a blank on the term, but they're anaerobic and aerobic, just like nematodes and bacillus are the type of bacteria that are facultative. That's what it is, facultative.
Ooh.
They're facultative. They can live in, and those are usually the good ones because, like a bacillus is a good bacteria that's facultative because they can live in our gut where there isn't much oxygen, and they could also live in the soil where there is a bit more oxygen.
So facultative meaning that they can both, like you said, live and not live, or live in oxygen and live without oxygen.
Yep, exactly.
Very good word. Awesome. Yeah, I mean, I've yet to see a tar. I think if I see one, it would probably be my favorite just because of how cute they are. But I'm gonna say I really like the rotifers. I want to see one so bad stick out its like mouth and like have like the cilia just and suck in all those. I haven't seen that yet. I only see the ones like that like kind of launch their neck out. And,
yeah, that's when they're not eating, they just kind of like swim around and stick around. I was actually watching one for like 30 minutes just waiting for him to open his mouth because I wanted to get a video of him in the dark field because my thumbnail is a clip from a really good video that I got a rotifer in the bright field when he was opening up and eating, and you could see everything swirling around him and everything, and actually got it focused enough so I could see those cilia moving, creating that vortex. Yeah, and that's the thumbnail for my channel. But I was trying to get him in the dark field the other day. I spent like 45 minutes and I maybe got something decent, but I don't think it was nearly as good as the bright field one that I got.
what do we have to do to bribe these microbes to do what we want them to do?
Yeah, I know. Like, open your mouth, do something. Go over here. Move when you're standing still. Hold still so I can see your mouth when you're moving sometimes.
So we have Lunar Soul. He's asking, what is in the center again? Is that an aggregate with the black outline? I believe, yeah, I guess he's talking about that next to the amoeba. So Lunar, what's up, Lunar?
It's getting slightly aggregated with whatever this is, but it's an unaggregated mineral or salt. And salt being in the loose term of salt, not meaning, table salt or whatever salt is, but, anything that's just a neutral soil substance, meaning, has a positive, has a negative, and it's bonded to something, just making it a neutral mineral.
And salt could be anything. It's like gypsum could be considered salt. It's like it's calcium sulfate. So you figure the calcium has double positive and the sulfate has what, double negative.
Yeah.
Right. So it seems like Lunar's talking about he said the one that looks like I'm sorry, Lunar, I don't mean to assume your gender. He said the one that looks like a pill. So that's the amoeba.
That's the amoeba. Yeah, that's the testate amoeba.
Testate amoeba.
Matt, would you consider the soil to be more of a playground or a war zone?
I like to think of it as like Yellowstone Park. You want to make it with all that biology, and if you take out the wolves, it's gonna go awry. You bring it back, and everything mellows out and starts thriving again. That's the way I like to think of it.
That's a really good description.
That is a great description. So I kind of want to continue on with that, but to do that, I was watching your video of your garden, and I was watching the last garden video. And also, by the way, your Swiss chard looks awesome. I was like, you made me so hungry when I was looking at that. But I thought it was really interesting. That like you were documenting like the different beds, and like you're looking under the microscope of the different like the different areas in your garden. and what you saw in there. So like what month and what area do you think did the best in your garden?
Surprisingly, that Swiss chard bed, and it was the thinnest bed. Like I bought a bunch of local soil just from a cheap local place that, you know, wasn't tested, didn't really know what it was, but it was local. And, you know, I mixed in some Fox Farm and didn't have my compost set up yet. So next year I would definitely mix in my own compost and, you know, at least top dress that. But and I kind of did the same thing to all the beds, but I was surprised that one was the thinnest. And there's here in New England, we have a pretty hard layer of clay, you know, maybe depending on where you are, anywhere from 6 to, you know, 12 inches under the soil. And this spot where I made it was probably only 6 inches under the soil, and it had like hard clay. So I'm really surprised that that bed, like the basil in there really liked it, the strawberries really liked it, the Swiss chard really liked it, and the peas really liked it. And then the bed that I actually thought I put more work into, being the cherry tomato bed, would actually, you know, didn't perform nearly as well as my cherry tomatoes last year. So it's kind of interesting. And, you know, looking at the soil, you know, after the year went by, the cherry tomato, you know, was obviously the poor looking soil, both under the scope and just, you know, looking at it in 1x with your eyes.
If you read the book The Martian, the guy in there, Mark Watney, he's like trying to grow plants on Mars, and he actually mentions in there that microbes move up through the soil. And I'm not sure how true it is because it's, you know, a fictional book and everything, but maybe those microbes like being close to that clay possibly. But
well, there's a lot of nutrients in the clay, but they're locked up. So maybe just the fact that I was putting on a lot of you know, compost extracts and, you know, other you know, biological amendments on it, that they just access that, all those nutrients locked up in that clay quicker, you know, started making it plant available. Who knows?
How do you like to amend your soils?
Mostly top dresses, you know, worm castings is a great thing. You know, just, I mean, compost is the #1 go-to. My teacher actually never stops talking about kale. So I'm right here on the ocean. So I've been experimenting with getting kale, because the main thing he says about it is it just has an abundance of nutrients. Actually, most seaweed has, you know, has a little bit of everything. So it's a great thing to, you know, put in your compost or, you know, top dress with a little bit. And there's a bunch of kale products coming on the market that are interesting just because it has just kind of a, you know, has all those micro and, you know, macronutrients.
I tried
I
using
remember.
kelp. Sorry, go ahead, Rob.
Go ahead. No, you go.
No, I was going to mention I had tried using kelp in compost before, and it just smells horrible whenever it's like hot. Like hot kelp is just like the most disgusting thing ever. I don't know if anybody else has any experience with it. I think the best, yeah, the best smelling compost ingredient for me is malted barley.
Yeah.
And you can get it in like a crystal malt or like a roasted malt, like you make a porter or a stout out of, and like that kind of starts smelling nice, like a bakery.
Yeah, that's another top dress that I like to do is the malted barley, you know, like the dried seeds, and like crush them up and just kind of sprinkle them right on the top.
Definitely.
It has you know, all those good seed growth hormones in there.
Yeah, like the whole malting process. Sorry to cut you off again, Rob, but like the whole malting process is like they get the seed to sprout and then they like dry the thing really quick so that it kind of preserves whatever chemical, you know, makeup is in that sprouting seed. And that just sounds really good for like a soil application too.
Sorry, I just wanted to say, I know I saw your video of your compost tea, and I thought that was really interesting. Do you want to talk a little bit about that? How did that was that like a good inoculation technique to add beneficial microbes into your soil? Did you see any difference if you applied it?
Yeah, everything I put it on seemed to really like it. And it was just kind of an experiment. It was just one of those things that I just kind of, yeah, I'm pretty sure that video was the one that I kind of threw the whole kitchen sink on it. I'm just like, all right, this is good. Let's put a little bit of that in. Okay, this is good. Let's put a little bit of that in. And then since I have a microscope, I could just look at it before I put on the plants. Okay, we don't see like one species dominating or any kind of crazy infestations or anything weird. So let's put it on the plants. And yep, that was pretty much how that one went. But yeah, I think we had some crushed barley in that one. We had oyster shell, we had compost, we had kale. I put quite a few things in that.
Yeah, so that is actually what I was most interested in, is that oyster shell. Royce, do you have any like input into like adding oyster shell for a compost/compost tea?
I definitely used oyster shell flour before. From what I understand, it's just like really finely crushed oyster shell. And from what I read, it releases calcium a lot quicker than other calcium-based amendments. Another one that's like kind of similar is called aragonite, and you can get it in Pennsylvania locally here, probably like the whole Northeast. But it's just like really old deposits of like seashells that are kind of ground up, and you can even see like seashell bits in there. And that one's supposed to be really highly available calcium also. I would say it's like the premium version of just using ag lime or using gypsum.
Now, kind of sticking with the aquatic nature, how much of a similarity do you find aquatic microorganisms to soil microorganisms?
Very different. Very
Really?
different. Yeah, yeah. I looked at a product that's actually made on Cape Cod, pretty close to me in Massachusetts. And they make it all from called fish brew. And there's all these things like antoms and all these. I was asking the creator about it. Actually, I've been meaning to email them back and talk to them more, because they're I guess one of the girls that work for him is a aquatic biologist. So yeah, and it looks completely different. I mean, there's a couple of similar shaped bacteria, but yeah, it's quite different from what you see in the soil.
So if I'm not mistaken, I thought that soil microbiology derived from like oceanic microbiology. I mean, obviously evolution and millions of eons, but happened.
Yeah.
But so it was a little surprising for me because I'm not too familiar with it. That you said it was very different. Sorry.
Yeah, well, I don't know. This is just the theory. This is just me thinking, but the water biology was advancing for a huge amount of time, and then it split from that and started the soil. So the water biology already had, I don't even know how many millions of years leg up on the soil biology to once it started advancing. So now we have soil biology, but water biology is still millions of years more old than that split once it came onto land. So it's just a little bit more advanced, a little bit different, and yeah, they move around a little bit different.
Yeah, of course. I mean, yeah, millions of years of evolution will do that.
Yeah.
Let's see. So do you want to go around this slide? I know it looks like it's
yeah.
drying up a little bit, but I want to find a fungus. I want to find that fungi.
Yeah. Well, here's these are spores right here. These two little football things, those are fungal spores. That's why I stopped here when I was scanning around. But this is not a fungal strand. That is a weird double cell wall. That's kind of interesting. But that could be a little root tip or something, or just organic matter. Come on, fungi. Always when you, oh, here we go.
Yes.
There's a nice little fungi strand. So you see the separation right there. And yeah, we see another hyphae right there. And it's just holding another one there. It's just grabbing on all those aggregates and breaking them down.
Wow. So there's a lot to unpack here. I how would you describe like what is a fungal hyphae?
So it's just like this network of mycelium, essentially, just, mushroom and most of it's actually interesting how much there is beneath our feet that won't even like sprout up a mushroom, or it'll, sprout up a mushroom once every, I don't even know how many years, but not all the time. Like it'll just maybe pop up one here. But other than that, it's living, it's doing its thing, it's spreading through the soil. Like what's the thing Paul Stammen says is like there's like miles beneath, like just under your foot at any given point in time. So it is pretty crazy. And yeah, it's a network, it can deliver nutrients to your plant from other places that the roots can't reach, and it moves things around, and it's just another part of the soil food web.
So I guess so the hyphae itself isn't necessarily like that single strand. The hyphae would be considered like just like that branch, the branches.
Yeah, and if it wasn't shaken up in a slide, this would go on for who knows how long, connect all sorts of different plants together. And if it's, a mycorrhizal fungi, it could actually inoculate and go into the plant roots. And then once it forms that inoculation with the plant roots, they are now symbiotes and working together.
Right.
Nature's internet. I just saw that there.
Yeah.
Micro 44, nature's internet.
Yep.
Yeah, I mean, I don't think I was reading about how there was a study being conducted. I don't remember exactly what forest, but in that forest they were finding that the trees were linking with the mycelium, and they were kind of like using the mycelium to send signals to other trees to kind of let the tree know, oh, there's like a disease on me. Maybe like start preparing yourself to combat that.
Yeah. There's a decent amount of science showing that very well might be true. I don't think it's quite proven. There's also, pheromones and whatnot that they'll release. That's like almost a way of releasing a scent. A plant will start producing a reaction to protect itself from a pest. The other plants that are next to it smell that, essentially their organic pesticide that they're creating themselves. And then the other plant will start doing that same thing. Be like, oh, my buddy's over here protecting themselves for that. I might be next, so I better do that too. I think it's more often the smell, but at the same time, I would not be surprised in the least if they actually talk even deeper through the fungi.
So Matt, you're blowing my mind here. You're telling me that not only are plants omnivores, but they also can smell.
Yeah, or sense the smell, or detect the chemicals on their leaves. I don't know quite exactly how they do it, but yeah, they do. They can, when one plant does a reaction, which you can smell, you can smell the difference of a plant when it's trying to fight off pests compared to when it's not. And yeah, I think the plants can totally sense that in their own way. Whether it's just that chemical blowing over and landing on their leaves and then being like, oh, this is from a plant for this reason. And so I'll do that too. Who knows? But
So
they're more intelligent than we think. They're definitely more intelligent, plants that big, than we think.
I know. I mean,
or than we know.
What do you think about the current research being done on like plants and stuff? Like, do you think that we're at a point where like you're kind of happy with like how much research is being done, or do you think like there's just way more room to continue to grow and learn about plants and their interactions in the soil and mycorrhizae?
I think there's a ton of room, and we're just the ball's just starting to roll in that direction. And yeah, once it fully starts rolling that direction, I think it's gonna we're gonna see some interesting stuff.
So can you explain, kind of going back to the fungus? Um, can you explain septas? I know in your live stream you use that a lot, and I just want a little bit of a definition.
Yeah, it's just these little, basically it's the separations in the cells of the fungi is what it is. And how you identify a nice fungi is what nice equal ones. They should all be uniform. So like this, there's one right there, then there's one right here, and then there's one right there. They're all pretty much the same distance. We got a branch coming off. So that's a good sign that it's a beneficial. You know, we don't know if it's going to be an endophyte or actually inoculate roots or anything like that, but it's not a bad one. It's not going to it's not a mold, it's not a actinobacteria, because you wouldn't see those if it was bacteria, if it was a bacteria strain. Yeah, so it's it's gonna. Oh, and the interesting thing I just learned in the class the other day is fungi process their food on the outside of the hyphae. So they're actually, around it, processing on the outside. So that's another reason why they're really good for the soil, because they're doing all that chemistry outside of the actual strand.
So I think that's
like micro changing pH, releasing, cations or whatever else they do to unbind things. They're actually, doing that in the soil around them, not digesting internally.
Interesting.
Now, I remember you talking about the different colors when you're looking at these fungal strains.
Mhm.
Is a darker color better, or this one looks a little lighter and more clear?
Yeah, darker colors better, but it could also, you could have a clear one that is just a young hyphae. It just started growing, so it doesn't have any nutrients in it, so it appears clear, and it could still be a really good one, but just hasn't filled up with those, organic acids yet, those humic acids. So makes it kind of difficult. So yeah, that's why it's just, it's another way of looking at things. Just like you go out and look in the soil, in your first couple of years of gardening, you might not notice all that much, but then as you get wiser and get more experience, you start noticing all these little things. And I feel it's the same way with looking at the microscope. And main reason why I started the channel, because I found so little out there, and I just wanted to see more and gain more experience. You can find 10,000 people walking around their garden with a camera, but very few people, looking at their soil under a microscope.
Yeah, I mean, there is definitely a need and a want for that. Sorry, I just looked at the chat. Someone said BRB, changing pH. Now, whoa, all right. So all that going on, that's because the microscope slide is drying, right?
Yeah, I think things are drying and pushing things in.
These microbes are like
Whoa.
sliding to their death, right? Like outside this little slide here.
Yeah, where can we go? Over here. Oh yeah. Oh yeah, I could do a quick oh yeah, there's the edge. It's coming in. But didn't find any big microorganisms in here yet. But we didn't look around all that much. But I did want to do a quick example of what dark field looks like.
Please.
And I'm gonna, well, yep, there, that's what's happening and pushing everything. See that bubble just drying and expanding?
I feel like whenever I see a bubble, it like breaks the immersion, because like it has such a distinct outline. It's like, what is that?
All right. I might have to go manual here, but almost there. Yeah, so this is what kind of looks better if I switch over. Switching my camera. There we go. Manual mode. I think I was Last time I was on manual, I must have been looking at stuff in the dark field because my settings are perfect. But yeah, so this is where you know really start seeing the humic acids and what the bacteria is breaking down really start glowing. And
Wow.
This is
everything
amazing.
kind of twinkling in the background is bacteria moving around.
What?
And
What am I looking at? I need this in VR like immediately. It's so amazing. You know, something that I heard on a podcast about soil aggregates is that in that aggregate, there's a much lower pH, just in that little tiny area. That makes so much sense, like for plant roots, because they can access the area outside with the higher pH and maybe different nutrients are there. And then right there next to that aggregate, there's going to be a low pH environment. It's going to be releasing a lot of micronutrients and things like that. It's probably easier for the plants to eat that in that way.
exactly, yeah, but like everything, all the biology will micromanage the pH. The roots at the tips will, put out exudates to change the pH and manipulate the pH in the way they want it. And yeah, so you can find a, way lower or like, usually it will shift between, 2 to 3 pH throughout the soil.
Interesting.
In
That's pretty
different
low
little
pH.
macro areas, and it's just, yeah, because certain elements take a certain pH before they'll become available, before they'll reduce.
Wow.
So when you don't know, when like if you can't identify something, what resources do you go to figure that out?
Usually, I mean, just start searching around, but sometimes it's disappointing because you get, pretty low res images most of the time. And then a lot of the times, like the microcosmos, I'll go on there, even though everything they look at is, in a lab. So it's not exactly, in the soil or anything that like you would see in the soil, but it's still the same microorganisms. So when they go through and identify microorganisms, they're one channel that has, I mean, they have a ridiculous microscope. I think it's tens of thousands of dollar microscope. I think they're rocking over there. And yeah, they definitely have the best imagery. And as far as, just, and they put the name of the organism up on the screen, so that's a good way to tell. Other than that, yeah, most of the time when I'm Googling and finding images on, Google Photos or whatever, what have you, it's tough. It's not easy, but that's what I usually go to.
And that's just because of the lack of resources, right?
Yeah, like all the images on Wiki, like it's, like that testate amoeba that we were just looking at before, like we didn't even work on zooming in or doing anything, and it worked way better than it looked 10 times better than the testate amoeba you'd probably find on Wikipedia.
Well, it's a good thing, Royce, we're training a neural network to be able to recognize different like aggregates and microorganisms.
Yes. I could see coming in from the bottom right of that slide, it's like a black hole coming in here. Like the one from that movie Interstellar. There it is. Coming up.
The bubble's coming in.
Looks interesting too.
Yeah, it's the black hole.
It's the edge of the universe for microbes there.
Yeah.
What would you say is like the black hole of the microcosmos? Like what organism or what what kind of abstraction?
I don't know if there is one. I like to look at it as a more positive view, but I guess well, as far as the anthropods are signs of it going anaerobic, so you never want to see a lot of those, or basically, I guess the black hole would be seeing like an infestation of omycetes, or just like actinobacteria just all over the slide, like everywhere. Like, just like the entire thing was covered with a spider web of that would probably be the worst. That would be like, holy cow, like what pH is the soil and what is going on here?
How would you change like a compost or a soil that was dominated by the actinomyces or something that was bad like that?
Mhm. Well, the actinobacteria like a lower, I mean, like a higher pH. So you could try lowering the pH, and then yeah, you would have to, just you'd have to get the good ones in there. So like EM1 has 53 some odd good, microorganisms in there. So I would start with, trying to get the good guys back in there by putting that on, and also, mycorrhizal inoculants and stuff like that would also help your situation.
Hmm. I
Now,
use a product. Sorry, go ahead, Rob.
go ahead.
Oh, okay. I was wondering what you thought about this one product. I like to use it whenever I like dip just like small plants that are growing in like little tiny pellets or like peat plugs or something like that. It's a product called Recharge.
Yeah,
And
is that AEA?
this is Recharge is from a company called Real Growers, and I'm on their
No, that's
website.
Rejuvenate, I'm thinking of. Yeah.
That sounds right. Where's the thing? They have like a listing of different stuff that's in there, and I wanted to get your perspective on like, are these kind of products worth it and useful? And so the one uh, is called Glomus aggregatum or something. Does that sound Glomus? Anyway, there's like 4 different glomus. Is that a bacteria?
It's got to be. I'm not good on the actual Latin names of everything. Usually I'll
Okay,
just, cut down to the as easy to remember as possible, and then if I need to, I'll go look that stuff up.
gotcha. It looks like this one has like 4 different glomus. It's got a Bacillus subtilis.
Yeah, that's bacteria, bacillus. Yeah,
Okay. It's got a Bacillus megaterium.
yeah.
Sounds
There's,
big.
there's
Got-
so many types of bacteria. We haven't even identified them.
Wow. Anyway, this product also has humic acid, molasses, and kelp in it too.
That's
So it
pretty good.
sounds
Yeah.
like microbe food.
Yep, yep. Molasses to keep them going. But
Now, how does molasses keep the microbes going?
it's a food source for them. But one thing, like once the molasses run out, then they got no more food. They're gonna start dying off. I don't know too much. Like EM is supposed to be stable. Like there's no food in it. It's supposed to be in a stable form. Like until you open that bottle, like it has a shelf life, so to speak. So I'm not quite sure how they managed to do that. I know they're all put together in a lab. Um, selected in a lab, put together in a lab. But then you can extend EM. So how you would extend it is you would, add molasses and seal it off so there's no air in it, and let all the bacteria and the microbes in there eat the molasses until it returned to its normal pH, and then it's ready to use.
So EM is effective microorganisms.
Yeah, it's made by a company called Terraganics. They're one of the longest in the game, have a ton of research into it. And yeah, so that's one that I had to get because my teacher talking about it, and yeah, them just being in the longest ones in the game. And then there's another product that I was trying out. I'm interested in like the kefir products and the Yahweh, and that's a good, but basically it's all bacillus form, bacteria, or as to the best of my knowledge, that's the main type of bacteria that you're going to see in kefir or ferment or something like that. Some spirula too. Definitely can be some spirula in there also, but yeah, there's so many ways to ferment and grow. Or
Now, can you touch on kefir? I like just learned about that, and it kind of fascinates me.
yeah, it's like I don't understand exactly what it is, but it's the whole cheese thing. It's like a cheese, it's basically milk and the byproduct of fermenting it. So what the bacteria does to the milk and, breaks everything down. Not quite exactly sure, but I just know it's a milk product and it has something to do with fermenting. And if you get it, you get a bunch of bacillus and good bacteria that are good for your gut and our plants. That's about the extent of my knowledge on it.
It sounds like kombucha that's made from milk to me.
Yeah, pretty much. Yeah, it's a good way of putting it. I think there's some slight differences and stuff like that, but I've heard people, actually making cheese and then using all that extra liquid as lab as labs, for kefir products and then or just labs or ferments or what have you.
So when you change the subject a little bit, but when you're adding these inoculants and these different products, you're introducing, I guess, foreign bacteria and fungi and just foreign like organic matter in general. Do you have any concerns about that when you're like adding these foreign like products into like your native environment?
Yes and no, but essentially they'll regulate themselves. As long as, if you added something foreign to dirt where there was no biology, that would be a bigger concern than if you already had a decent, thriving native biology. Chances are the worst thing that can happen is the microbes that you just paid for die. And still, that's even those dead microbes are still going to be processed and nutrients for the soil, even if they do die. So it's not 100% loss, but usually that's the worst thing that can happen.
Now, how would you like mitigate, say you have non-beneficial nematodes, which are the root eating kind, right?
Yeah.
How would you try to like scale their population back if you observe that?
You can buy like just predatory nematodes. So that would be the main thing. I was actually looking, what was the other way? There's a couple of other things. There's actually some plants that release exudates that are anti-nematode, marigolds for being one. They're kind of an anti-nematode or root feeding nematode. But there's also a fungi, I forget the name of the fungi. But there's also a fungal inoculant that you can use that it will actually tangle up the nematodes. So there's a couple of different things. So it's pretty much just all about, identifying your problem. And usually it's going to be not just seeing one or two things in the slide. It's going to be an abundance. You're going to see your slide loaded with one thing and nothing else when you do have a problem. And then it's just looking to see, okay, what eats that, or what don't they like? What pH do they like? Are they anaerobic? Are they aerobic? And then sort of, going through your resources and your books and figuring out, different things to combat them.
Makes sense. I mean, I'll give you a little case study for myself. So when I was first composting, I was composting a bunch of kitchen scraps, trying to add, I wasn't adding as much brown material as I probably should have been. But I was I got a sample of my compost, put it under the microscope, and I saw like, it was hundreds of nematodes. At the time, I didn't really know how to identify them. I was just more fascinated by, wow, there's all these wiggly worms going around. But over time, checking that compost pile again, there weren't as many. So would you say time itself too is a factor in trying to mitigate like population sizes?
Yeah, and food source abundance, like kitchen scraps are pretty abundant in food. So, it's definitely, you can see a party going on there, or, once everything evens out, they'll kind of spread themselves out and other things will start coming in to eat that food. But yeah, the main thing is, I wonder if there was multiple types of nematodes or all one type. But usually when those party parties happen, they're over quick, but you want them to happen in your compost. You don't want them to happen in your soil, because that could be a very drastic change. And if that's going on around the roots, that's why I've heard some like bukashi stories where people have buried a bunch of bukashi and it goes anaerobic and it being a bad thing. And I've also heard some stories of, ridiculous success for a bukashi. And I think it's all about what happens. And if the right organism gets a hold of that bukashi and blows up, it could cause a problem. But they might not even be a bad organism. It could be a beneficial nematode. It could be but there's just so many of them that it causes a problem. You want that nice diversity of, and web, not a big party of one organism, because that's not going to last long. It might crank out a bunch of nutrients, but it's probably going to be too much nutrients. It's probably almost going to be toxic at that point. And yeah.
Now, I'm going to quote you here. You said that you want your microbiology to be like an old growth forest. Can you elaborate on that?
Yeah, I said that a while ago, and it ends up turning out to be, depends on the plants. So if you're growing fruit trees or blueberry bushes, they're a much more advanced soil species. So yeah, you would want it to be like an old growth forest, being fungal dominant and very diverse. And then more like your brassicas and stuff like that, you kind of want more bacteria in there. You want it not so advanced. Like brassicas probably wouldn't do as good as I thought they would when I said that in an old growth forest, So just for an example. But to go to the next level is now all our crops are basically bred for the soil that they're growing in. So what if we could breed crops like brassicas and simple, lettuce and stuff like that to grow in a more advanced soil that's fungal dominant? Would they be 10 times more nutritious and grow even better than we ever imagined? It could be.
So do you think that, where's the balance between trying to inoculate the soil to have the microbiology at a certain threshold versus I'll say like kind of genetic editing of the plant to be like able to live in a certain climate / soil condition?
Well, I think saving seeds is huge, because that genetic information that they gained over growing in their climate, and you're going to use those seeds in that same climate the next year, I think that is huge. Does that pretty much answer the question? I was going to go make one more point on that, but I forgot my train of thought.
No worries. I mean, yeah, it did. I was just kind of throwing out like a little thought experiment, just also for the viewers themselves. If anyone has any comments, leave it down below. Yeah, thank you.
So yeah, in that same light, I think we could advance our plants to be much more nutritious and thrive in more advanced soil with more biology. I think we've been moving the other direction, and we've seen for a fact that our nutrition from what we get in the supermarkets is way lower than what it used to be.
Now, do you think people should be growing more like obviously the answer would be yes, because it's a good answer to say yes to, but like realistically, how do you see the future how do you see future agriculture? Do you think it's going to be more localized, where you have small farms providing for local communities, or do you think we're just going to keep skewing more and more towards large ag providing all the food for everyone? What is your dream for that?
I would like to see more local. more market farms, more local, and more just a smaller community sustainability as far as that goes, and not have to ship all the food all the way across the country all the time. I think that would be a huge thing, just economic wise, for every you know every little part of the, no matter where you live, no matter what town. I think that's key, to have that self-sufficiency on your own food production. Well, let's see, other than that, I kind of see it going to the thing where it's going to get forced that direction, because we're running out of these fertilizers. We don't have enough inorganic fertilizers to keep on doing what we're doing. So eventually we're going to be forced into the direction, and when people, hopefully it's going to be before it's like, what do we do now? And then, so eventually they're going to have to turn that way, one way or the other, and start looking at the biology to taking care of their plants instead of dumping all these chemicals on there. Because you look at the forest, nobody's putting any fertilizers out there. Big old trees, they grow every year, looking healthy. I have to trim them back out of my driveway. They grow faster than my garden.
Very good point. Very good point. And I think maybe if like the fertilizer prices keep on climbing like they do, it's going to become economically viable for farmers to start using different microbial products and different practices, maybe that they find work in their situation. And economics is probably going to drive that, just because of cost prohibitive fertilizers and things like that. Because farmers do have to make a living, and farmers definitely try their best to grow the most food possible with the least cost. It may be just having more information about how to do that using microbes, because obviously there's trillions of different microbes in the soil just helping out with feeding plants. And so getting them to figure out the right practices in their area is probably huge. So it's channels like yours are definitely going to help them, and hopefully more people do some work like this and figure out what are the best practices that provide the best payoff.
Yep, baby steps. And once people see it working, they'll adopt it more, and then maybe we'll see this super large scale compost teas and compost extracts happening instead of all the spraying and everything. Because it would, I really think it might be harder in the first year or two, but after that the cost savings and yield increase are gonna speak for themselves. It's already speaking for itself, and it's just a matter of time before it really gets out there. Everybody's looking over to their neighbor's field and be like, oh, what are you doing? And it's just like, oh, haven't you heard of this regenerative soil thing? No, I haven't. Oh, well, yeah, check it out.
Get on board.
It's eventually, hopefully that's the direction we're gonna go. And
so I'll give a good example. I help on a farm in New Jersey, and they practice regenerative agriculture. They go off of community shares, I think is the CSA shares, community service shares or something. Anyway, but they got the farm, so it's about, how many acres do they have? I think it's about 70 acres, and they got the farm off of another farmer who used a lot of non-regenerative practices, lots of fertilizer, like just pumping the plants with chemicals and drugs. Pretty much.
Yeah.
But now obviously they're trying to gear away towards that. And I brought my microscope over to the farm to analyze the soil for them. And it was kind of sad, actually. Like the soil you have on screen right now looks a lot more diverse and full of life than what theirs did. I mean, it's still growing plants and stuff, but it just kind of goes to show you just how much of a detriment like those things end up having on the soil itself. I mean, the only thing we saw, I think, was just a little protozoa just flying around. It's actually really funny. When I was showing people, I was showing the one lady, and she saw the protozoa too. And I was like, see, I told you I saw something, because we couldn't find any life. And everyone else looked at us like we were crazy, because they're like, there's nothing there. What are you talking about? But I mean, my point is too, though, is that it just takes lots of time, especially doing it the way that we ideally want it to be done. Unfortunately, it is a long process, and there is a video coming, part of my book review series. I read a book called The Long Now, and it talks about a project that's really made to enhance people's scope of what it means to like progress and do things like over long periods of time. So we're not talking even years, we're talking like decades and centuries. And I think gardening and farming is like a long now project. It would make more sense for the people who understand that project. But pretty much just like you have to think long-term, long scale, especially when it comes to this stuff. Unfortunately, I mean, with technology, our attention spans are going, but I think gardening and farming is a very good way to practice kind of that sense of it's like a good way to discipline your foresight in a way.
Yeah, it's always, you always have a goal when you're looking at the soil under the microscope. You always want to strive for more fungi or more diversity or more aggregates. It's kind of fun in that way. And you never know what you're going to find.
So what do you find the most fun when you're analyzing under the microscope?
Definitely finding different critters, nematodes and stuff like that. Just looking around for them. That's probably the funnest part, because every now and then they jump out at you. You don't see one, and then all of a sudden it starts moving. It's like, oh, whoa.
Yeah.
Kind of make you jump in your seat every once in a while.
Have you ever gotten like do you get the heebie-jeebies whenever you're looking at some of these things? You're like, ew, like, it's kind of gross looking.
No, not really. Not like I would with a spider. Even snakes sometimes. Like, I like snakes. I'll pick up a snake, but I kind of pick up the snake just because it gives me the heebie-jeebies when it's, crawls on you or what have you. But yeah, nothing I see really gives me the heebie-jeebies that I've seen yet anyway. Maybe if I think about it, if I imagine being, if you imagined a rotifer or something being like our size, that's kind of scary and terrifying.
Yeah. Oh God, I don't even want to think of that. That's like that thing.
If your mind doesn't go there, it doesn't weird me out at all.
Do you see like even when you're like in normal life, like do you now have a different perspective of like the microverse?
A little bit, yeah. I definitely find myself looking around and just being like, it's usually when you look and you see that newly tilled field and it's just turning gray, being out in the sun, not covered. I'm like, oh, poor microbes. It's just that gray color is just like billions, or not even billions, it's trillions and trillions of just dead microbes.
I definitely agree with that sentiment. I feel like
the
that's
soil just eating itself. That's one thing I find myself looking around, and then I'm always happy when I see a field with cover and looking healthy, or a winter cover crop coming up this time of year. I was like, woohoo!
Do you have a preferred winter cover crop?
I'm still experimenting. I'm experimenting with I put some a mix of peas and clover and some winter purslane. Really hoping I'm rooting for the winter purslane. But the clover seems to be doing better, providing more cover at the moment in my garden.
Matt, do you think you could talk about what you think about tillage? Like, should gardeners ever till their home garden? Does it have any place in gardening?
Only when it's necessary. Only if you're not going to be able to get a seed into the ground that's going to grow. I think that's the only time. The only other time, I think, like aeration, using like an aerating fork or something like that. They actually have some machines that will go around and lightly till like just the top inch or two, just to get the seeds in the ground. That would be the only thing I would say go for. Other than that, it's necessary sometimes, but usually only when you're first starting. And if you have like at your Texas or just have hard packed soil wherever you may be, or sandy soil, like you're gonna have to turn something in there to at least get your seeds in the ground and grab onto something. Other than that, I would say as light as possible.
Interesting. All right. So a lot of gardeners think that they should do it every single year. And maybe that's because it just gets weedy and like the ground doesn't look nice to plant seeds into.
Yeah.
What do you think about covers? Just like some people use like a black plastic or like a landscape fabric. What's your opinion on covering it with some kind of fabric?
I used it. It works. I mean, there's no weeds. I didn't have any weeds in my garden when I used it, but I feel there's a baking problem. It's black. It heats up, and from what I've heard, you'll have higher soil temperatures than compared if you use the just say a mulch or straw or something. You have much higher soil temperatures using the black mulch, and that would be my. I mean, the black plastic. That would be my main concern. Is it getting a little too hot for those top roots on the top of the soil, and that's that's that whole rhizophage section. And if you kill the biology in that section from getting it too hot, or only kill all the ones that don't like it hot, you're just lowering your microbial diversity.
Good point.
Do you know any like ideal temperatures that microbes like?
Not really. I know. I mean, the only thing I know when it comes to temperatures is like composting temperatures. You want to get it up around 150, 160 to kill the bad ones, and then you got to let it cool and get back down to ambient temperature before it's good to be used in a hot compost method. But that's really the only temperature thing that comes to mind. I think it all
So,
depends on the microbe and what they like.
hmm.
But they're robust. They can be frozen, thawed out, still be alive. They could be way down in the earth crust where it's heated up to super hot temperatures, still survive.
Have you ever taken samples like so typically I'll take a sample like about an inch to 2 inches deep. Have you ever taken any like deeper than that in the soil?
I usually do like a 6 inches, and I try to take a little bit of soil from every inch. So I use an apple core. I put the apple core all the way down through the soil, and I take a little bit from everywhere, and then kind of mix that up when I do a sample. I feel that's the best way to get a good average,
Yeah.
And but other times I'll see some cool stuff on the surface, or some fungi or whatever growing, and I'll just take a little pinch right from there. Take my scooper right out and just be like, that looks cool. Grab that, see what that looks like. So now I know what's going on in that top layer. But if you want to get an overall feel for the soil, I usually go down 6 inches and try to get a little piece from every inch.
Do you find any patterns like or like do you see any reoccurring, I guess, themes of like how many microbes there are in certain depths of the soil? Like, is there an ideal, like, do you see like one stage of the soil having the most activity?
Yeah, usually the top 2 inches usually have the most. That's where, especially when you have straw, leaf, mulch, or any type of mulch on the surface, they're actively coming up there to break that down. As long as it's not too dry on the surface, once that top layer dries out, they head down.
Yeah.
or die.
Most
Oh.
likely the second one, because microbes can't move too far. Like very, very short distances, right?
Some can. I think we'd be surprised. I don't know. I'm guessing there, but a lot of the bacillus can swim pretty quick. Actually, I forget what was but there's one type of cilia that can actually move faster for its body size than anything else on the planet. I forget what like streptomyces something, or it was strepta something. And they could build up all their energy and go like 100 times their body length in like a split of a second. It'd be like us, jumping a football field in 1 second.
That is so wild. Interesting.
Would it happen to be the Spiro? Oh goodness.
Yeah, it was a tough one. I remember it being I'm never going to remember this one.
Spirostomum. That's where we're going to go.
I think that's it. I think that's the one.
Found
Very
in lake
cool.
pods, moves using tiny hairs. Yeah, the cilia. That's amazing. It can move it can shorten its body by more than 60% in more than a few milliseconds, going from 4 millimeter flat ribbon to the shape of an American football without using any kind of muscle tissue. Interesting. Well, I know what I'm reading tonight.
Rob, I was wondering if you were willing to pull up your soil sample.
Sure. So I know, Matt, we didn't really look. I mean, we were browsing around in your soil. I don't know if you had anything that you had in mind that you wanted to show us. I mean, now would be the time to kind of take us through.
Yeah, that was about it. Oh, we're all dried up on this slide here, unfortunately, but it's the dried slide. But well, pretty much when I go in, I like to go in at 100. Look at the aggregates, look around for any big organisms going around there, and the main thing that you're going to see is, I mean, you're looking for what's not there, and you're looking for what's in abundance. Those are going to be your biggest clues. So if you see one species taken over, you might have a problem. If you don't see anything, you know what you want to add in there. And those are the main things that you can take away from looking at your soil under the microscope, in my opinion.
Cool. Well, I want you, Matt, to judge me now about how I'm going to go through this process of analyzing soil. Okay.
All
so
right.
bash on me if you want. I have tough skin. It's all good if you just tell me I'm stupid. I'm just kidding. But all right. So here we have a soil sample from my garden, my garden bed. It has a top layer of mulch. I just cleared it out of all the vegetables. There's actually still a single little finger eggplant plant that is hanging out in there because it's still growing. But besides that, it's a fully done and exhausted garden bed. So I'm curious about how this is going to go. But I have my little test tube here. I have a pipette. Now, Matt, do you do a 5-to-1 ratio or I'm sorry, dilution ratio, or what do you do?
I like 5-to-1.
How I feel.
Just because I like to see more stuff.
So I'm gonna get now. Can you explain the different dilution ratios? Why would you prefer 5-to-1 over say 10-to-1?
Counting. So if you want to count, you definitely want to go 10-to-1. Otherwise, you're just gonna have that much more stuff to count if you go 5-to-1. If you're just doing video and just documenting the video, and that's what you're saving, I say go 5-to-1 because you're just going to see more stuff, makes it easier, don't have to go through as many slides. But yeah, when it comes to, I've tried to do the counting process from what I know off of it, and I haven't figured out a good way to do it. But yeah, just imagine counting all those little bacteria down there. So if you do a 10-to-1, you're going to have a lot less to count per quadrant, is essentially what I gather from it.
So it's more for the quantitative analytics. that
Yeah,
you
for
use.
quantize, and you always have to do the same ratio to base that data off anything else.
All right, so I would say, I mean, I count that as 5. What do you think? You think I diluted that too much?
Looks pretty. Is it? Did you shake it up already? Or
Not yet, not yet.
not yet? Yeah.
So we were talking about this earlier. I call it the Elaine Ingham shake because she's the one who popularized it for all the internet soil geeks. But yeah, you just do a little rock back and forth motion very lightly. Now, Matt, you mentioned that you're not crazy about letting the soil, or I'm sorry, the samples settle as much as Elaine Ingham is. Can you elaborate on that?
Yeah, because I found, well, one thing I learned from Crescent Soil and Scott Scams is that nematodes like to swim down and sink. So he found a problem where on a farm where they are watering from a bottom drawn tank, so all the nematodes would go into the first where they first watered, and none would get to the back sections. So, and I tested it out myself just with a sample that I found. And if I do the typical shake and then just let the sediment, any rocks, because you don't want a big pebble or big piece of sand in your slide, because in your slide is going to be a couple millimeters more off of there and hard to see and yada yada. So just let the sediment settle for like a half a second, go in quick. And in my experiments, I find way more nematodes that way. I was just looking at for nematodes when I did it, and I did three samples doing each way. I found multiple samples on all 3 slides that I went in quick, and I found only 1 nematode per slide on the 2 that I let it set for 30 seconds, and 1 slide I didn't find a single nematode. So definitely seems that certain organisms will either sink or float. So I like to get it stirred up, just let it set so that sediment gets out of there, and then grab it as quick as you can.
Awesome. Thank
That's
you
my
for
opinion.
explaining that.
Anyway,
How many shakes do you normally give?
usually 30 seconds.
All right. Well, I
So,
think
you've probably done enough at this point. Plenty, I would say.
there is such a thing as too much shaking.
Well, you're just gonna break things up too much. You don't want to break up the hyphae. You want those nice long hyphae. Sometimes I'll shake less just to try to find a nice long hyphae if I'm want to take a picture of some fungi or something.
Makes sense.
I try to go in right next to the roots and just be extra gentle with it to try to get nice long fungi strands, if that's what I want to take a picture of. But other than that, if I'm just doing, you want to be the same every time if you want to document or try to quantify some data. You always want to do it the same way. But if you're just having fun, do anything you want.
Okay, so this is the
See the difference.
scariest part about the microscope is the slides, because now I'm missing the
Yeah.
cover slide and I have no idea where it went.
Oh, that's the worst. That's the worst. Don't crush it.
Yeah, oh, I found it. Okay, we're good. Lunar Soul says no on the shake. Yes.
I stepped on one in my rug and I vacuumed the heck out of it, and I still think there's glass in it. Scared to step on that spot with my bare feet. Just tiny little glass shards.
That sounds awful.
I was shining a flashlight on it, and I kept vacuuming, and I was still seeing things shine. It crushed into sand-sized glass shards that were just stuck in my rug.
All right, so I'm setting this up now. Let me make sure. Light on. Why is that like that? Oh, it's because of that. Okay. I am placing the slide on the microscope stage.
Do I need to stop sharing my screen, or can you just take over?
Yeah, you can stop sharing yours for now, and then I'll
Alrighty.
little meta right there. Just cover your face with mine real quick.
Mhm.
Okay, now let's start zooming in. Here we go. Okay, cool. Now, Matt, I don't have as good of a setup as you, but I'm gonna try my best. Move that out of the way. Screen two. Wait, no. Camera. All right. Can you guys see what I see?
Mhm.
Great.
That doesn't look too bad.
Now, I'm immediately noticing. Oh, I gotta figure out how I can do this. All right. Is that right there a little fungal spore?
Focus a little bit more. It kind of looks weird shaped. Not an aggregate, is it? But what magnification are you at right now?
Right now, I am at it looks like crap. I always forget. Yeah, it's the 40x.
40x. It's kind of large. It's probably, and see how there's something in the middle of it. Usually
Right
fungal
there?
spores are all even. That could be a cyst. So like some sort of organism cysted up. So it forms this little cell wall around it, and this little environment for it to live inside until it wants to jump out.
Now I will say actually, because I just looked at the eyepiece. This camera zooms in a lot more than what like the actual eyepiece does. So this is more than 40x. This is
a little bit more. So it could be a fungal, but seeing how there's that shape in the middle, that could be something in there. Usually the fungal spores they have that nice uniform cell wall and even shape throughout the middle, but
all right. Well, let's continue browsing.
could be. I think it looks more like a cyst than the spore.
Now that looks like a piece of
Yeah, it's a little clear hyphae of something.
tiny one. Yeah, I wish the quality was a little better, but let's keep perusing. Maybe we'll find.
Yeah,
Oops.
it gets the job done. What's that little camera shooting? Is it just
It's from Amscope.
HD? Is it? I mean, is it just does it just capture HD video?
Yeah.
Is it like 1920 by 1080?
Actually, no. It's way worse than that.
Oh, it's like 720.
Yeah, it's like 720.
Mhm.
Yeah, I mean it was a little $50 thing, but
Yeah. I can,
no lunar. It's bright field, not light field.
yeah. Mhm. But you look
So-
like you got a nice fat piece of a fungi strand right there. Over to the right.
That dark kind of.
Yeah, that dark little short strand, and
Yeah.
another one over to the side. Those look like fungal. I think I can see a hyphae in there. I'm not sure.
Yeah. Here, let's
I see a little.
get a little zoom. Boop.
Yeah, there's one.
It looks like a beard hair. Like if you shave when you have stubble, and there's just like a tiny thing. And
Yeah.
you're saying that's a piece of
Yeah, it's a
fungal
piece of a
hyphae.
fungi.
Let's see.
Wow.
Make lunar happy and maybe make it a little dark field.
Mhm.
Actually, this microscope, the camera doesn't catch the dark field as much. At least when I do my
Oh wow, look at them. They're all
yeah,
moving.
not too bad. Yeah, see, it starts glowing a little bit from the dark field. You usually get some neons in the fungi hyphae. It's kind of a way that you can tell if they're alive or not.
Wait, how can you explain that a little bit more?
They'll glow neon when they're living and not as much when they are not living.
I'm currently at a what is this? This is 100x. Someone asked in the chat.
So Matt, would you say this is a good soil from like a microbe perspective so far?
we're in there pretty far right now. We're only really seeing one. I've only seen one shape of bacteria swimming around. I would like to see more aggregates, but I mean, it's not dead. And we are seeing pretty much a little piece of hyphae on just about every frame we get to. So that's a good sign.
And actually, what's really interesting is this is more hyphae that I'm seeing now than when I did when I first analyzed the soil bed. So that is a good thing. What is that little? That's a
something moving around in there. Could be probably a fragilate.
Yeah, it looks like it could be. Because you can tell like by how silly it's moved. Ooh. How silly
they
it's
usually bumble.
Yeah.
Sometimes you can see their tail.
It's going crazy. Now, what exactly, like, are these bacteria? They're just like hanging out and just eating pretty much. That's all they do.
Yeah. They just kind of wiggle around until they bump into something they like.
All right. So this right here is an aggregate, correct?
Yep.
And can you just explain again exactly like this is pretty much the food source of the bacteria, and the glow is from all the humic acid that they release?
Yep, exactly. Yep. So they release those to change the pH and break down the mineral and make it available for them and also plant available.
Yeah, you see all the little bacteria just like around it. So cool.
I was doing
I was like,
go ahead. Sorry.
No, go ahead, Bruce.
Okay, thank you. I was looking around the internet to try and get like a scale for how large these things are. And I was looking and it said, let's see, bacteria is 1 to 2 microns wide by 5 to 10 microns long. And for perspective, I wanted to know like how big is a clay particle, like the smallest type of like aggregate in the soil. Not aggregate, just like, soil particle size. There's sand, silt, and clay. And it said that the clay particles, the largest ones are like 2 microns. So in terms of width, they're just about as big as a bacteria, the largest ones. So clay particles can get pretty small.
Yeah, they can't get, but I mean, they're bigger. I mean, most of these larger things that we're seeing are, smaller than a particle we can see. Like, that right there might be close to sand size. But if you put a piece of sand on there and you're at the 100x 1,000, yeah, it would fill the whole screen. You put one piece of sand on there. Like
So,
a grain that you could actually
soils,
see with the eye, it would, it would. We'd be looking at a piece of it.
soils are pretty small. That's really interesting.
The silt's the smallest, right?
It's sand, silt, and clay. Clay being the smallest. And I read one time that if you took a teaspoon of clay, of pure clay, it has the same surface area as like a football field, which is crazy to think of.
Yeah.
What?
Yeah,
that is kind of crazy. So yeah, probably these little pieces here are little, silt and clay.
I also wanted to know like how big is like one ion of like say calcium or potassium. So this is like 1 unit of a plant nutrient, and it's basically so like an ion would be just like an atom of some element, pure element, but it's like charged. And so that's measured in something I haven't used before called an angstrom. And it said an atom is about 1 to 2 angstroms wide, and a bacteria is tens of thousands of angstroms wide.
Yeah.
So if you just zoom really, really far in, like you could probably never see one using like a regular microscope. I wouldn't imagine.
Yeah, no, I think you need electron or fancier or
something. Quantum microscope or something.
Yeah.
You think that's plant matter?
Above it, yes, but that one right there kind of looks. No, that looks like more like mineral that's getting chewed up. Mm,
And then that piece right there is that
yeah,
would be
like that's plant matter.
Yeah. Now I don't mean to go back on aggregates because I'm sure you already answered this question. But like the aggregates, like what are they like? What is that's just all organic matter kind of clumped together, right?
Well, it could be all different stuff. One aggregate could be a clump of this and that, and the other one could be, one could be phosphorus, one could be copper, one could be zinc, one could be iron. They can,
Cool.
and probably most of them are compound of quite a few.
So that's an aggregate forming right there, right?
Yeah, they're just starting to hop on there and eat that.
Let's zoom in a little bit further if we can.
Aggregate is like a microbe city. It's just a
Mhm.
bunch of them all together around this really tasty piece of dirt.
Yeah.
They're living, they're dying, they're creating humic compounds, and all of their exudates and their goo that they create is just gluing this thing together. And the fungal hyphae are in there linking them all together. It's like a city.
Yeah.
Awesome.
I realize the eyepiece is a little dirty when I tried going what magnification is this 400x. So I gotta clean that. So that's not the best, but
Yeah, a little deep in there. The more you magnify, the less focal depth you have. Just one of those optical, physics things that you can't really get around
yeah,
with
that
light.
physics.
Right in the middle there kind of looks like a fungal spore, that little brown football.
Yeah.
Yeah, right there. We'd have to see it in the light field. Could just be an aggregate, but
I can
a
move
little
it.
piece of
Ready? Light
little
field.
piece of clay. Oh yeah, yep. That's definitely a fungal spore. It's the right size.
Oh no. Oh, I'm so sorry, Luna. I didn't see that you wanted to zoom in on the wiggle.
Mm.
So I guess the fungal spores they have more of a football shape, not necessarily like a more oval, not spherical in terms of like straight circle.
They can be round. Like I've seen ones that look like little something on a like little balls on a Star Trek or something.
So like that
An alien
next to
shape.
the hyphae or to the fungus, do you think that's a fungal spore?
That could do a little bit more focus. Yeah, it looks like it's got a cell wall. It's got that outer wall. It's uniform. Yep, I think that's a spore too. Oh yeah, and now you see how it's glowing a little bit like that. So that's alive.
Wow, that's so cool. Rob, how about you talk about like what you actually did to the soil, how you grew, what did you add to the soil to actually make this happen?
Oh man. I have to like look at my notes. So I started off the raised bed. I actually have pretty good soil just to begin with. Royce can attest. We we got a nice soil test from Agger and did an analysis on my soil, and it was pretty good, but there was still a lot to add. And what I added was gypsum. I know Royce, you were already bashing peat moss earlier, but I did add some peat moss. I just wanted to try it because a little bit new, but what else did I add? I added lime. I added limestone. And honestly, that was about it for now. I didn't really do a lot. I kind of want to see like how things would grow naturally. My first real summer of growing, and it actually ended up really good. I mean, I had a full successful garden. Go to the Agger Instagram. Actually, I don't even think we have a good picture of that. Go look at the first year experience and you'll see the garden and all the plants that I planted. And they all grew really well. I think the only one that didn't grow was the Hummingbird Falls flower, but that wasn't planted inside that garden bed. And that was just a plant that required a lot of high pH, and my soil did not meet that standard. And I wasn't trying to change the pH too much for just that plant. Oh, what is this over here?
Yeah, I was like, who just came walking in the top left over there? That's what I was looking at.
But I also have a cover of mulch. I'm sorry, I have a cut like my mulch or my cover is straw. I use straw. I'm very much a straw person.
That thing just ate something.
So
What do you
like
think that is, Matt?
I think it's well, it just broke off in the white piece that I thought was dragging it, but
Right. Is that a spirilla?
yeah, I think it was just a no, they're more like little tiny snakes. But it's probably a little cilia, flagellate of some type. And I think he was stuck to a couple of little pieces of clay.
It's a wiggler, right?
Yeah, and then he shook them off.
Hey, look at him. He's like eating.
Hmm.
No, unfortunately, we can't get much closer. My eyepiece is kind of dirty. The camera itself isn't that great. Oh, there's another one coming in the right.
Hmm.
Um,
Yeah, what do they look like in the bright field?
yeah, sorry about that. Let me look. Oh, can I
Let me see.
lower? Oof. Yeah, the bright field doesn't look that great with this camera.
Yeah, it's a little. Gotta get a little darker. Got a little side shading now to see him, but
Ooh, it's a lot of activity.
yeah, but there's one moving like that. Yeah,
He's carrying something. You see it.
yeah. Or it could just be just stuck to him. But looks like a flagellate. Look round.
In one of the shorts we have on our videos, or it might not be a short, it might just be a shorter length video, but I noticed there was a nematode that was stuck to a piece of like mineral, and I felt so bad because it was trying its hardest to get off. It just couldn't, and I was like watching it like a maniac. Like I was just like how can I help it? You know.
So I see them that like that a lot that seem like they're stuck. But I kind of feel like I think they actually will stick their tails onto something and hold on as they flail about and try to grab stuff.
Oh,
Oh, yeah, that's more like it's probably a bigger ciliate, not really bumbling. That was swimming more.
now protozoa. Maybe I'm mistaken because I know I
Proto-
mentioned that earlier. I thought like they were the super fast boys that are going around. Like
Yeah.
that kind of reminded me of one.
Yeah.
What is a protozoa exactly? Because I think I have my stuff mixed around.
Yeah, I get mixed around. I still need to look at notes every once in a while, but it's a small type of cilia. I mean, they usually have just one cilia, and they have like a tail, and that's what they use to swim around, and they move really quick with it. They sort of fly around. But I forget the classes, which one comes first, because it's tough because it's not like there's just one protozoa. There's hundreds of different types of protozoa. And does it fall into the ciliate class? Does it fall under like I think forget if protozoa and then ciliates or it's vice versa. I always got to look it up.
Which is fine. I mean, that's why the internet is the internet.
Once I look it up, 20 to 30 times, I don't remember.
It takes me 40, so you're good. So what are your thoughts here, Matt? What are your thoughts of my soil?
Yeah, it's looking pretty decent. Definitely seeing a lot of clear stuff. So, that's just, more bacteria, more stuff, but I'm not seeing any weird strands, I'm not seeing any actinobacteria or any signs of it, going towards that higher pH 7 and up. So it's probably has a pretty good pH level, I'm assuming.
What
Since
is
there's
that
none
black?
of those in there.
Oh, wait. I thought that was a nematode.
Yep. Always use more diversity. It'd be nice to see, like a couple of bigger organisms in there, but we're not going, usually I'll start and I'll literally scan the entire slide at a, higher or lower magnification, however you look at it, but zoomed out more, and I'll just scan the whole thing for big organisms and then go in and start looking at what I want to look at a little bit closer. But
no, I'm really interested in what this thing is. It looks bigger.
I think it's just because they'll clump up the bacteria. That's when they're conjugating.
So I'm trying to look at it with.
So sometimes you can actually catch them splitting too.
Hmm.
It's easier to catch like different types of small amoeba and stuff like that splitting, but every once in a while you can see little clusters of bacteria
All right, well, let me take a
doing their
broad-
thing and multiplying.
and let's see if we can find any interesting
Yes, now we're out
like that. What is that? No, I don't think so. Now, what do you find to find like the macro microorganisms? Is it easier to see them in bright field or dark field?
Well, the bigger ones either or. I find with my setup. But as far as one tough thing with my camera, I haven't been able to find any get a way to dial in the bright field over 100x. So if I want to go into 400 to look at bacteria,
Hmm.
a little circly swimmer. Right, little protozoa, flagellate, or some type of like that. But the dark field is nice for kind of anything living tends to kind of glow. Like all those strands, that's actually a really nice big long looking hyphae. Right over there on the right. Yeah, middle now. Yeah. That's a big one.
I'm not really seeing the septa here though.
I saw one, but yeah.
Oh,
Sometimes,
there you go.
oh yeah, you see, definitely see the septa usually a lot easier in the bright field. Yeah, but they
That is really long.
and that's a big one. So that's like what, 4 or 5 micrometers wide. You take this, the smallest bacteria you see and estimate that being 1.
Very
You know,
cool.
it's probably, yeah. That's a nice looking fungi, and another one right there too.
Now I want to ask you, Matt,
That was a
how,
good high
how do you
five.
not get lost in the sauce, as a way of me saying it, in terms of like when you're like analyzing the soil? I find that I can sometimes like really get like discombobulated and be like, where even am I?
Just by looking at the slide, I usually have a system, so I'll start top left, and then I'll kind of go back and forth like rows. And anytime I see something interesting, I'll try to remember where it was so I can go back to it. But yeah, as soon as you start getting a little lost, I'll be like, wait, was I just going right or was I going left?
Matt, what do you think about looking at compost under the microscope versus soil? Do you see way different things from the compost?
A little different, but no, not really. Ultimately, you kind of want to say the same thing, because what Elaine said, one of the best plants she's ever grown is 100% organic matter, pretty much all compost.
Now, is that a cyst?
Um, Kind of looks like it. Well, doesn't look very uniform, but it looks like there's something in the middle and a cell wall around it. Yeah, I would say so. You don't see those internals that you usually see on an amoeba. You usually see a lot of round nodules, and
Yeah.
that just kind of looks like some little critter is bundled up in there in the middle.
Hmm.
Sometimes you'll find them and you could actually make out like a nematode in there. And
Really?
you actually see the nematode.
So it'd
I
be like a
saw
baby nematode?
or just one that just didn't like the environment and decided to go into a little protective cocoon, essentially, until the environment got better.
All right. I think because I don't want to hold you too long, Matt, but I'm gonna quickly pull up a compost slide, and we can just
Alrighty.
analyze real quick what compost versus my soil looks like. Now, this would be compost from Royce.
Yeah, I would definitely say a good job on the fungi in there. Definitely seeing quite a bit. There's very few frames where you don't see at least a little piece of a fungi.
Just trying to quickly peruse through to see if we miss any. Yeah, I'm not seeing any big guys, which is unfortunate.
Yeah, the only thing would look like where could use a little bit more aggregates, but it'd be nice to see at least, one or two bigger microorganisms in there. I would say that's the only thing missing from your soil food web at the moment.
All right. Royce, I'm actually missing your compost. I don't know where it went.
Oh,
Sorry.
it's all good. I only gave you a small sample.
Where'd it go? All right, well, I'm not gonna dwell too much on it.
Hey, Pop.
All right. Well, I think we'll wrap things up here. But I want to ask you, Matt, before we wrap this all up, if you could see anything change in the current industry of agriculture and gardening and just growing in general, what would you want to see?
The corporate greed go away. I think that would help the most.
Careful, you might get someone coming to your house tonight.
I mean, they could do better, but they don't.
Good answer.
All right, Matt.
Yeah.
Well, it was a pleasure having you on our podcast. I'm very happy that you were down to do it live with us. It was the first time we did it, and it felt like it was a success. Thank you, everyone in the chat. Bless up with that. You guys were amazing. I really appreciate the questions and the engagement that you guys had. But yeah, besides that, Matt, I mean, right now I'm going to give you the floor. Shout out everything and anything you want to shout out to the void of the internet. And yeah, your time's now.
Yeah, well, just the one thing I wanted to follow up on the corporate greed thing is more so to the point is I want to see the farmer making the profit, not the corporations would be the better way to put it. And I think that's going to be really done by, using biology, because, they can't really patent that. And if we all just figure this out together, and there's so much more to learn, and I'm really excited for, databases and stuff like that being created. Matt Powers created the Our Soil database, where we're all going to be able to upload our microscopy and our chemical soil samples and our SAP analysis. And, it's just all about learning together and sharing our experiences, working and figuring out what works in our zones, and sharing that information so everybody else can learn from it as well.
Well said.
Very well said, sir. Very well said.
And I kind of want to touch on real quick about that, like the community, almost like open source for gardening. I know in the tech world, that's a very big term, open source. I mean, right now it's fairly popular with the AI, such as Stable Diffusion was open source, and that's taking off. So yeah, having like more of an open source feel in the gardening industry and tech industry, which is also, I mean, something that Royce and I definitely want to help with as well, I think is the key. So, but again, I mean, Royce, do you have anything to say?
Excellent podcast. I really couldn't agree more from the farmer's standpoint. Just like Matt said, it's important to get more tools, more data sets into the hands of people that really need it. Yeah, I think the farmers could benefit, gardeners could benefit, and it's just going to grow such a better society if we have this awesome food that's really cheap, it's cheaply grown, these microbes, and better soils. So
And
+1
health.
for that.
All right, guys. Well, thank you so much for watching this podcast. Leave a like, subscribe, and let us know if you have any questions. I mean, we have live chat now, but we're always going to be able to see it later. And actually, Lunar, if you're still here, we have a video specifically for you about the cation exchange. So stay tuned for that too, my friend. But yeah, again, thank you all so much for watching. Thank you, Matt. Go follow, go subscribe to Shooting the Soil. Amazing content that he produces. And that's it. Take care.
Until next time, friends.